Hello, everyone. Thank you for attending today's webinar, Metabolites and Safety Testing, MIST, Analytical Strategy, presented by Frontage Laboratories. I'm Ricardo Castano, and I'll be moderating this webinar. Our speaker today is Philip R. Tiller, the Senior Director of Frontage Laboratories. You can read his full bio on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. This webcast is being streamed to your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Okay, now let's begin. Dr. Tiller, please go ahead.
Thank you, Ricardo. Good morning or good afternoon, everyone. Thank you for joining us. I hope all are well in these difficult times. What I want to do, first of all, is just go briefly through what I plan to discuss today. So let's move to my next slide. Okay, so what I plan to do today is first of all give some background to MIST, where it started and what it is we're asked to do by the regulatory agencies. The bulk of my presentation, about 80%, will actually be on addressing the analytical challenges involved in conducting a MIST study and going through the scientific underpinnings behind how we have constructed our approach to address what is required of us from MIST. And then finally, a few slides discussing a case study as an example. So MIST started based on a pharma meeting that occurred in 2001. This is a technical industry group, and they had a meeting where they were discussing the impact potentially of circulating metabolites. And then some of the leading lights in that group, who are actually co-authors on the paper here that was published in 2002, they actually addressed some of their concerns in the way they thought it would be best to address them, and they first used the term, this is the first recorded use of the term metabolites in safety testing. After that, the FDA generated a draft guidance that was published in 2008. That was followed up by the ICH issuing their own guidance in 2010. And then the FDA issued their final guidance in 2016, which they've just updated this year, March of this year. Next slide, please. So here are two quotes from the FDA guidance issued this year. We encourage identifying any differences in drug metabolism between animals used in non-clinical safety assessments and humans as early as possible during the drug development process. Discovering such difference in drug metabolites late in drug development can cause development delays. So some of the background to that, I think the FDA had experience with a couple of instances where human metabolites were first examined in the human mass balance study in phase 2, and there were some concerns there that resulted in the development delays for that clinical program. So they're recommending that we do this type of assessment earlier in the clinical phase to ensure that there are no clinical development delays. And then addressing how they want us to address this, generally metabolites identified only in human plasma or metabolites present at higher levels in humans than in any of the animal test species should be considered for safety assessment. Such metabolites are called disproportionate drug metabolites. Human metabolites that can raise a safety concern are those present at greater than 10% of total drug-related exposure at steady state. There is another statement in their misguidance. They also make reference to glucuronides as being metabolites that are of less concern. Could we have the next slide, please? So this is a quote from the ICH guidance, and I won't bother reading it all. Again, I've highlighted in light blue the bit that's most important. Again, 10% of total drug-related exposure and at significantly greater levels in humans. This would be the level at which point there would be some concern about the safety coverage of a metabolite. So the way that has been typically interpreted to date is that if your human exposure of a metabolite is twice that observed in one of the two safety species, that is acceptable. So we get a little bit of leeway there. Next slide, please. This is a flow chart you find in the FDA guidance. So what we want to be doing is going down the left arm, which is we find no drugs that exceed the 10% of total drug-related exposure. Therefore, we don't have to do any further evaluation. Based on our experience of more than 100 MIST studies, that is a rare occurrence. We almost always find at least one metabolite that is greater than this 10% threshold set. So we would expect to find a metabolite that passes this 10% total drug-related exposure threshold. And now we need to determine, is it formed in one of the animal test species, and is it formed at levels equivalent to or greater than the exposure in the human? And if the answer is no to both those questions, then you need to look at doing some non-clinical testing with that drug metabolite. And our goal is not to go down that path, but that unfortunately occasionally is where we get to. Next slide, please. So what are the basic steps of a MIST study as far as the FDA and the ICH guidances are expecting? So they want us to select samples from steady state. And what we at RMI and Frontage have always recommended is that we take the human dose that's closest to the anticipated therapeutic dose and compare that with the animal safety assessment doses at the known adverse effect level dose. That way, there's a best chance of getting coverage and exposure margins by that comparison. So we need to identify all circulating human metabolites to be able to effectively determine there is no metabolite that's of concern. And then we need to determine which of those metabolites are greater than 10% of total drug-related exposure. And then we need to demonstrate that those metabolites that are greater than 10% TDRE, that are also present at similar or greater exposure in one of the safety species. And then 0.5 here is what we don't want to get to, but we will occasionally, if we don't have that coverage, we then need to look at further safety studies. And the bulk of the FDA guidance addresses 0.5, assuming you will have a problem and what to do with it. I will be addressing points 1, 2, 3, and 4, hoping we don't get to that point. Next slide, please. So early, the DMPK analytical community came and started coming up with ideas on how do we address this guidance. So this paper that was published in, let me remind myself, 2009, so this is the year after, from the BMS group, they're suggesting that there were 3 approaches that we could take. One is using a human mass balance study with carbon-14. The other is using an accelerator mass spec study, also using carbon-14. Both of those are somewhat expensive, and so the question is whether that amount of investment in a program this early in its clinical development was a question. And the other suggestion they suggested was using accurate mass technology of non-labeled studies. And that has been the approach that has been taken by the DMPK community. Could I have the next slide, please? So the same group published this paper in 2014, where they describe a tiered approach, where we use a research-grade assay to identify and quantify metabolites to address the misguidance, and then if there is any further need to quantify metabolites in ongoing subsequent clinical studies, then you use a qualified or a validated assay for that. And so that is the approach that the community has typically gone for, where we use high-resolution mass spectrometry in a research-grade assay to conduct our studies to comply with the FDA and ICH guidance. Could I have the next slide, please? So there are several challenges in using a research assay to conduct these MIST studies. So we need to identify all human metabolites. We don't have a radio tracer, which, in the early 2000s, this was the gold standard in doing metabolite ID. And this will be in plasma, so this is a complex, relatively complex biological matrices. So that's the first problem. So could we go to the next slide, please? And let's address this, identifying all metabolites. Next slide, please. So we need a method that will identify all expected metabolites. This is metabolites we could anticipate by looking at a molecule and suggesting you could add oxygen here, you could have demethylation there, and that constitutes about 95% of the metabolites we would observe. We also need to be able to identify the unexpected metabolites. These are metabolites due to things like ring contractions, where something unusual happens and it's not something that's easy to predict. Again, this represents about 5% of metabolites. And then this method has to be both capable of providing qualitative and quantitative measurements. Could I have the next slide, please? So HRMS has been determined to be the best solution, and I want to do a little bit of background as to why that is the case. So let's look at some basic physical chemistry here. If you look at the nominal mass, that's the third column here, this is the mass you effectively can measure using a triple quadrupole or an ion trap. The column on the far right is the exact mass. And you look at nitrogen and hydrogen, their exact mass is slightly higher than the nominal mass. And you look at oxygen, chlorine, fluorine, and sulfur, and their exact mass is slightly lower than the nominal mass. So we call nitrogen and hydrogen mass sufficient, and oxygen, chlorine, fluorine, and sulfur mass deficient. So this actually is important going forward. So this is how we're able to differentiate between our compound of interest and the biological matrices. Next slide, please. So here, let's look at a hypothetical xenobiotic on the right versus a hypothetical peptide, a thiogly on the left. They have the same nominal mass, but their exact mass is substantially different. It's actually different by 32 millidalton. Now, take into consideration that your modern accurate mass device is accurate to within plus or minus 2 millidalton. If we had both these compounds running on our system, we would be able to differentiate them simply based upon the mass of the ion using accurate mass. And just some background, each double bond you incorporate into a molecule decreases the mass by 16 millidalton. And then if you incorporate oxygens or halogens, this also decreases the fractional mass or the mass defect. That's the number after the decimal point. So what does it look like when you try and make these comparisons then? Could I have the next slide, please? So this is an extracted ion chromatogram from a hepatocyte incubation of glyburide, where we're extracting the mass for the oxidative metabolite. Here we're using a mass window of 0.7 Dalton, so we're replicating the quality of data you would get using a triple quad or an ion trap mass spectrometer. And then to identify which of these peaks are metabolites and which are just matrix interferences, we would have to interrogate all of them, do MSMS on many of them perhaps, to identify what are actually drug-related species. In contrast, if we could go to the next slide, please. This is the same data where we're using a 20 millidalton window, which is what we typically use when we're extracting ion chromatograms from high-resolution data. And you can see that there are 5 oxidative metabolites, just based on the mass of the parent. We have been able to eliminate all of the interferences simply using the accurate mass data. For your information, the 4 metabolites on the left are from the cyclohexyl moiety, and the one on the right is actually from the 2-carbon linker in the middle of the molecule. So the point here I'm making is that accurate mass data allows you to differentiate matrix ions from the ion of interest, drug-related components, just based on the mass of the ion. Next slide, please. So how do we use this HRMS data to find the metabolites we're interested in finding? So the first step is we extract chromatograms for the potential metabolites. Now, you can do this manually. We would recommend using a computer program, since they're very good at doing something as simple as this. You can see from the table on the right, this is showing the exact mass shifts for potential formula shifts and the table we use contains more than 70 potential formula shifts. So in this instance, we're taking Crixivan, we'd take the mass of Crixivan, and we'd apply these mass shifts from all of these and find which chromatograms actually contain a peak. But that's not all. That's only the first pass of trying to find potential metabolites. Could we go to the next slide, please? What we also do is we actually conduct, theoretically, ND alkylations, OD alkylations, amide hydrolyses, things of this nature on the parent molecule and get what I call pseudoparents and apply this list of potential metabolites to each of these. As you'll notice looking at the exact masses of these ions, the mass shifts quite substantially in these differences. So we're trying to make sure we don't miss anything as a resulting from a secondary or tertiary metabolic process. So having done that, we'll get the data we see in the next slide. So here is an example chromatogram from data we've acquired. This is actually rat hepatocyte data, and this is a sum of all the metabolites found using those extracted ion chromatogram approaches. And we have 18 metabolites, if I'm correct. So this is a combined extracted ion chromatogram of all 18 of those metabolites. So this approach, where we extract chromatograms based upon what we think might happen, applied to the parent and to the pseudoparents, will find 95% of metabolites. That means there are still potentially 5 metabolites that we will miss if we're only using this approach. Now, we can get to this point using a triple quad or an ion trap. It'll take longer because the data they deal with is more complex. But we're at the point where we've only identified 90%, 95% of metabolites that are there. So what do we do? How do we get data on the 5% of metabolites? This is really where accurate mass data sets come into their own. Next slide, please. In 2006, the BMS group produced this paper where they introduced a novel approach to leverage accurate mass data, which they referred to as mass defect filtering, to identify these unexpected metabolites. Next slide, please. It's based on the observation that when you look at normal biotransformations like deethylation, demethylation, or hydroxylation in the case you observe in this table, the fractional mass change, the change to the numbers below, after the decimal point, are relatively small. So we see 31 for deethylation, 16 for demethylation, and 5 millidalton change for hydroxylation. So how do we use this mass defecting? They described mass defect filtering, and so I'm going to spend a few slides describing how that works. Next slide, please. So we're going to start with the exact mass of the drug of interest. So we're using Crixivan here. It has an exact mass of 613.363. Next slide, please. So this is a graphical representation of a data set. So these blocks here are representing all the ions we would see between mass 613 and mass 614, for example. So we would actually have there a mass at 613.001, 613.003, 613.004. Okay, you get the point. I won't go on. There will be something in the order of 800 or 900 separate ions observed in an accurate mass data set across that one AMU period of mass spectral data. And so when we typically use a total ion chromatogram, we just select one mass from that block. But what we do with mass defect filtering, if you could go to the next slide, please, is we apply a filter. So the filter, this window is about 35 millidalton higher than the exact mass of interest and about 50 millidalton lower than the exact mass of interest. So this orange block here represents that filter. So the block in the middle, around 613.363, shows us the area we would be collecting data from that one AMU. But then we collect the same window of data at 612, at 614, at 611, 610, 6.9. So we're extracting a 85 millidalton window across the mass range. So remember, we had about 800 or 900 data points there. By doing this, we are eliminating 90% of the data points that are irrelevant to the mass of interest to us. Next slide, please. So we apply that mass defect filter to the parent. We also apply it to these pseudo-parents as well. And we get this. Next slide, please. So the chromatogram at the bottom is the chromatogram we saw a little earlier. The chromatogram at the top is a result of these mass defect filtering. And you'll notice there is one new peak labeled new that we observed as a result of mass defect filtering that we could not identify based upon our expected list. And ironically, from this data set, which is why I'm showing it, of course, this is 19 metabolites versus 18. So that's almost exactly 5%. So using mass defect filtering will allow us to get to the point where we can identify 100% of metabolites that ionize. I need to emphasize that caveat. We are using mass spectrometry. So if you have a metabolite that does not ionize, these tools will not help in identifying them. Luckily for us, most of our xenobiotics ionize very nicely in either negative or positive ion. And so this works, broadly speaking, very, very well. Next slide, please. Okay, so we've been able to identify all of our human metabolites in plasma. Now we need to determine which of those metabolites are greater than 10% of total drug-related exposure. So we need to deal with ionization differences, and we need to deal with possible matrix interferences. So we know that different structures can result in differences in ionization. So how do we compensate for this? Next slide, please. So there are several ways. The best way is to use authentic chemical standards of metabolites. And you generate an equimolar solution of those, you run an LCMS run, and then you can simply compare the peak areas of metabolite versus parent and determine the differences in ionization potential. In reality, it's not often that we actually have authentic chemical standards of the metabolites at this stage in the clinical development of a compound. I have to admit, we have had one instance where we were able to get authentic chemical standards, one of over 100 examples. So this is, we call it our platinum standard. It's something we don't often get the chance to use. Next slide, please. So the gold standard, what is a really great way to do this is if you have a radial label version of that drug, where you do an incubation at increased concentration in microsomes or hepatocytes and obtain an in vitro sample that we then run and compare the mass spec data with the radiochromatographic data, and we're able to determine the relative mass spec response based on those two data sets. Unfortunately, radiolabel is not always available at this stage. So let's go to the next slide. What we sometimes end up falling back on is what we call the silver standard, which is the use of UV. So we would take the compound of interest, we would incubate it, again at higher concentrations, and use that incubation to provide UV versus mass spec comparative data to generate a response factor. And if none of these are available, if your if your drug does not have a chromophore, you then may need to use analogs or synthetic intermediates to be able to conduct something of this nature. Luckily, we've never got down to that level to date. Next slide, please. So we've been able to compensate for ionization differences. Now we need to deal with matrix interferences. How does matrix interfere with the study we're trying to conduct here? Next slide, please. So this is a schematic of the electrospray source, as it were. And What's happening is we're getting desolvation, we're getting accumulation of charges on the surface of the spray, and so you can get preferential ionization at that point, where phospholipids, surfactants will preferentially ionize, or there may simply just be so much material in your electrospray plume, too many analytes, it suppresses the ionization of the analyte of interest. Such interferences result in a nonlinear response, and so there's a risk of underreporting a metabolite's true exposure. So the little graphic bottom on the right is showing that we've suppressed the response, and therefore we're underreporting it. So how do we address that? How do we deal with that potential? Next slide, please. So Scott Obach's group in 2010 published this paper where they came up with a method that they were using to compare matrix samples from different matrices. Next slide, please. Let's give you everything at once. Okay, thank you. This approach has been coined matrix matching, where we take the human plasma and add it to control rat plasma. We take the rat plasma sample and add it to blank human plasma. So we now have two samples that have equal volumes of rat and human plasma. One has rat metabolites, one has human metabolites. You spike with an internal standard to compensate for variabilities in sample handling and mass spec response. You would then conduct an AUC pooling using the Hamilton approach, for example, and then you compare the two samples. So this approach is very, very good for comparing two samples. It does have, though, a major limitation. Next slide, please. Matrix matching may simply hide matrix effects. We haven't eliminated matrix effects, we've just normalized them across those samples. So for comparing two samples, that works perfectly. But if you're trying to identify the exposure of a human metabolite, we potentially compromise that and risk misidentification of humans that are greater than 10% of total drug-related exposure. So RMI and Frontage have updated and modified this approach, next slide please, to address the requirements of NIST. So what we do here, we take the human Cmax sample and we spike that into controlled human plasma, control rat plasma, and control dog or monkey plasma, whatever is your non-rodent safety species, and then dilute those further with those control plasmas as well. So we generate a dilution series, 2, 4, 8, and 16-fold, for example, diluted. We now use HPLC and HRMS to obtain data, and we determine the parent and metabolite response at each of those dilutions, and hopefully generate data much like the next slide, please. So we aren't really concerned about the slope of the curve. We're concerned that the curve is linear. If the curve is linear, what you've demonstrated is there is no matrix interfering with the ionization of the metabolites of interest across this dilution range. So the advantage of using the Cmax sample and diluting that is that will cover a nice range that's appropriate for the experiment we're conducting in an AUC pooled sample. If you see nonlinearity, you simply modify ULC gradient, so you pulled apart the metabolite and whatever the matrix component is that's causing interference, and you do that until you find linear response for all of the metabolites of interest. So having a linear response, we now are able to analyze, actually, could you hit the button once more? There's a comment at the bottom. So matrix matching may hide the problem, whereas we're eliminating matrix interferences by this approach. Next slide, please. So our approach to generate TDRE for metabolites is you determine the MS response factor to demonstrate linearity of MS response. And then we acquire, once we have that LC method defined, we use that LC method that we've used to eliminate matrix interferences to actually analyze the human, the rat, and dog plasma samples that we're going to use now for our TDRE comparisons. And we conduct that study in triplicate. By so doing, we get an average and a standard deviation for that percent TDRE determination. So that's the crux of the approach we take to generate robust total drug-related exposure numbers in our analyses. Next slide, please. So I wanted to go through a brief case study. So here, we had a client that came to us. They wanted us to analyze samples both from human and women at the anticipated therapeutic dose, compare it with the no adverse effect level plasma from monkey, and then plasma from both male and female rats that for doses that bracketed the no ALL was no longer available. So the compounding factors in this is that metabolites are observed in both positive and negative ionization mode. The greatest number of metabolites was observed in positive ionization mode, but the most abundant metabolites were observed in negative ion. And so we need to determine the percent TDRE across both positive and negative and come up with a combined percent TDRE to determine which metabolites are of concern, potentially. Next slide, please. Could I have the next slide, please? So we start with using the response correction approach both in positive and negative ion. Then we generate the dilution series to determine a linear response in positive ion, and it was linear. Great. In negative ion, the response was not linear. So we had to redevelop the LCMS assay to use in negative ion mode several times until we found that our responses were linear across that dilution range. Now we had the methods to analyze the AUC samples. So we now have a set of percent total drug-related exposures in positive and in negative ion mode. And fortunately for us in this study, the parent ionized both in positive and negative ion modes. So we were able to use the parent ion to compare those two data sets and determine an overall data set describing which metabolites were greater than 10% of total drug-related exposure. And we found several. Next slide, please. So what we take is what we do is we use the area ratio. That's the ratio between the metabolite of interest and the internal standard that is spiked into the AUC plasma sample when it is prepared, to generate area ratios at each of the data points. And then what we do is we normalize the area ratio for the most abundant human. So in this instance, for metabolite A, it was more abundant in the female than the male, B more abundant in the male, C and D more abundant in the female. So we normalize our area ratios to the most abundant human exposure, and then we compare the area ratios with the animal species. In this instance, you'll notice that all metabolites had exposures of greater than 10-fold in all instances. So we've now demonstrated that all metabolites observed in human that were greater than 10% of total drug-related exposure are observed at greater exposures in one or more of the safety species. Next slide, please. So our focus here is on generating a robust percent TDRE determination. So just to reiterate, we determine the metabolite MS response correction factors. We demonstrate MS response linearity. We analyze the AUC pooled samples from the human and the safety species by HRMS in triplicate, and our results we obtain from that is a TDRE average with standard deviation. Next slide, please. So I'd like to thank my former Merck colleagues who, when the guidance first came out, some very useful discussions, particularly with Tom Bailey, who was my boss at Merck at the time, and we've collaborated with him since several times. My partner in crime, Sean Yu, we've worked on this now together for more than a decade, and of course numerous clients, because the real data sets, the real samples are what provide us very interesting data. And thank you for your time.
All All right, thank you, Dr. Tiller. And now let's move on to the Q&A. There is still time to submit your questions using the Q&A tab to the left side of your screen. We have a lot of great questions already, and we will try to get to as many as possible. So let's see. Our first question for you, Dr. Tiller asks, it's a two-part question, really. It asks, in the generation of in vitro samples to determine MS response factors, what do you do when the in vitro system used does not generate an in vivo metabolite? And then how do you determine the response factor?
That's a good question, and that does occur often enough that metabolites that are really minor in vitro studies, such that you cannot really get a good comparison using UV versus mass spec, for example, end up being more abundant in vivo. So what we do at that point in time is we use a little bit of extrapolation, I think, is the best way to put that. So if you have an oxidative metabolite, it's very likely another oxidative metabolite, even though the oxidation site is in a slightly different part of the molecule, has not had a fundamentally different impact on the ionization. So we will make those assumptions. If this is an oxidative metabolite, then we will apply the response factor we've obtained from an oxidative metabolite that is close structurally to that. In the instance of a glucuronide, for example, we know glucuronides ionize potentially very differently than the parent molecule. Again, based on our experience, an average change is about a threefold difference. So we would, in this instance, if we don't have another glucuronide metabolite to use as a comparator, we would use a threefold difference as our response factor for that.
All right, thank you, Dr. Tiller. And just a reminder, if you guys do want to ask a question, feel free to do so using the questions and answers tab at the left side of your screen. And our next question asks, how much does such a MIST study cost, and how long does it take to conduct a MIST study in your lab?
Okay, the exact cost is dependent upon what approach we're using for determining the mass spec response factor. So if it's authentic standards versus radiolabeled versus UV. Broadly speaking, we're looking at something between the range of $45,000 and $50,000 to conduct such a study. A simple study will take about 3 weeks. If it's a much more complex one, like the case study where we have to do everything twice, it will tend to take a little bit longer.
All right. Let's see. Our next question asks, earlier in your presentation, you mentioned using NOAEL plasma samples from the safety species. What do we do if they're no longer available?
That's a good question. So that was actually part of the case study where the NOAEL rat samples were not available. In this instance, they used samples that bracketed it. Or you could actually conduct a satellite study where you dose to steady state at the NOAEL dose and then use those samples in the place of the safety species samples.
All right, thank you. Let's see. Our next question asks, have you tried data-independent acquisition workflows like SWAF for having a total picture and identification of your metabolites?
So the brief answer to that is no. We've tended to use the twofold approach I described here, where we use a list-based interrogation of the data set initially to find the expected metabolites, and then we use the mass defect filtering to actually collect information on the unexpected metabolites. Data-dependent acquisition is great for providing MS data on your unknown. In this instance, we're simply trying to identify which components in the sample are metabolites.
Okay, thank you. Let's see. Our next question asks, have you tried micro or nano flows to diminish the possible matrix effect, or other interference?
Actually, no, we haven't. That's actually quite a nice idea. My experience in micro flow work is that you tend to have a slightly longer dead time, but that's not necessarily an issue. It's just that we tend to use the same LC-MS approaches that have been used in previous studies for the client. So if you do discovery work and then you do some work in rat or dog in preclinical work, when we come to MIST, we like to use a methodology that's close so things tie together nicely. So there's no reason why you couldn't. It's just not the approach we have taken.
All right, Dr. Tiller, thank you. And let's see, our next question asks, any thoughts on the TDRE of phase 2 metabolites?
Okay, so the FDA guidance states specifically that glucuronides, and let's put acyl glucuronides to one side here, are of less concern. And so you can interpret that however you want to. So typically, my understanding is if you have a direct glucuronide that is greater than 10% of total drug-related exposure, that the regulatory agencies are of less concern about that. If it's a secondary phase 2 metabolite, they may be concerned about the potential concerns related to changing in degree of glucuronidation and whether the actual initial metabolite may be of concern. Getting to acyl glucuronides, they are specifically mentioned as a metabolite of concern. So if your metabolite is an acyl glucuronide, there is toxicity issues related to that. If you see that metabolite at the same or greater exposure in your animal species, then you would tend to be able to argue that you have safety coverage for it.
All right, thank you. And just a reminder again, if you have a question to ask Dr. Tiller, feel free to do so using the Q&A tab to the left side of your screen. And moving forward, our next question here asks, how do you know that all metabolites have been extracted equally across the species?
Good question. So what we do in generating the AUC pool samples from the human and the 2 safety species, we spike that with an internal standard. So we understand there's likely to be some differences in extraction and in mass spec response when you run different samples. The idea is we now compare the metabolite to the internal standard, and that compensates for differences in extraction.
All right. Our next question asks, can you please explain the process for glucuronides?
I'm going to have to answer. I don't know what you mean by that question. So could you answer that? Could you ask that question again in a different way so I understand what you mean?
Okay. All right. Good to know. Let's see if we can get the person who asked that question to rephrase it. But moving on, our next question asks, is there greater regulatory interest in specific metabolites based on structure and predicted toxicology risk?
The answer is yes. I've mentioned the acyl glucuronide, anything that has a known toxicological concern like an aniline. So yes, there are specific structures that are highlighted as to be of concern.
All right. And next question asks, do you have any comment on sulfates and TDRE?
So our relatively recent experience on sulfates is that they were a pain in the neck. That doesn't really answer what you mean by that. So as long as you're able to ionize the sulfates, you are able to determine its total drug-related exposure. My slightly flippant comment was based on the fact that we had some metabolites that were sulfates that were extremely abundant and actually were significantly more abundant than the parent drug of interest. So that's just anyway, hopefully that answers your question.
All right, thank you. And it looks like this will be our last question. We had a lot of great questions today, and we couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. So, Dr. Tiller, this last question asks, do you need to take plasma at multiple time points to analyze the metabolites, or just around the Cmax of parent?
Good question. So we use the Cmax simply for the linearity checking, but when we actually do our AUC pool, we pool across a time range. So typically we're using a 0 to 24 pool, so in which case we'll be taking plasma from each of the time points collected from pre-dose all the way up to 24 hours. In some instances where the drug of interest disappears very rapidly from the blood, we may actually do an AUC pool that is less than 24 hours, but that is dependent upon the PK data. So yes, we do. We take samples from multiple time points.
All right. Thank you so much, Dr. Tiller, and thank you all for attending this Fierce Biotech webinar and for submitting so many great questions. I'd like to thank Dr. Tiller once again for participating, and I'd like to thank Frontage Laboratories for presenting today's webinar. And remember, this webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.